Alright, so we are going to take a look at, in this video, trying to figure out what does it mean for recombinant offspring. How do we figure out their recombination frequency, and how do you make a map of those genes on a chromosome? So here we go. So we're looking at this gray-bodied fly here, and it has normal wings, and so it's got this B plus VG plus on the same chromosome. But it's heterozygous, and so it's also got B and VG on its other chromosome. And so you notice that these have been replicated, and so when they go through meiosis 1, we're going to have crossing over, and some of those chromosomes are going to mix up. And so some of those chromosomes, a small percentage usually, less than 50%, are going to cross over, and you're going to get some of this B mixing with the VG plus or the B plus mixing with the VG. In order to figure this out, you have to do a test cross with a homozygous recessive, and so that in this case is black body and vestigial wings. So all of their chromosomes, no matter if they cross over or not, are all going to end up BVG. And so you'll see that down here at the bottom, every one of the flies are going to end up with that. But what you're going to get is you're going to get all four possible combinations of B and VG, and the pluses are going to end up in the gametes, but two of them, the ones that got mixed up, the B plus and the VG, are going to be your recombinant chromosomes. And so that's going to show up right here on this one and on this one. And so you'll notice, and the way you'll be able to recognize this in the problems is that it's not going to come out 1 to 1 to 1 like we would expect. Excuse me, I underlined the wrong one. But it's going to come up in the black body vestigial wing and the gray body. These ones are the normals. They're going to look just like the parents, but these other ones are going to be in a smaller percent. And so the way you're going to figure this out is you're going to add up your total offspring, and so that would be the totals of all of these, and you put that at the bottom, and then you'll do the number of ones that were the smaller numbers. Those are going to be a result of recombination, and so all you do is divide those up, and you get the percentage of 17% in this example problem. And so what that means is that recombination frequency is a result of the percentage is the same as the number of distance between the genes on the chromosome. But you need to remember that if it's less than 50%, it's on the same chromosome. But if it's greater than 50%, that means it's on a different chromosome. Basically, they may be on the same chromosome, but you're getting the same results as independent assortment, so it doesn't really matter. You can only map genes that are having a recombination frequency of less than 50%. Here's another example of this. What we're looking at here is C and SH. Long story short, independent assortment, this would be a test cross for heterozygous with a homozygous recessive, and you'd predict 25% for all of them, or 1 to 1 to 1 to 1. But in reality, you can see these two are going to look like the parents, and you're going to have the most of them. In this case, I think it's 97%, and you're going to have 3% recombination frequency, which is going to mean those two genes, the C gene and the SH gene, are a distance of 3 map units apart. And so the reality is that you're going to get some problems, and these are going to be mathematical in nature, and you're going to have to kind of think your way through them. And so the first thing that I would do is take a line and just draw it across your paper. And what you're going to want to do, you can see these numbers represent the recombination frequencies between these 4 genes here. Okay? And so it doesn't really matter what they're called, but they're just letters, and we need to figure out how do they fit together on this. And so what you want to start with is the biggest distance. And so you can see that right here. VG and B are 19 map units apart. And so I'm just going to make a little notation. Put VG there and put B on the other end, and I'm going to write the number 19 at the top. So I know those are a distance of 19 apart. Now I'm seeing that VG and CN are 9 apart, and RB and VG are 16 apart. So most likely RB is going to be somewhere over here. Now we can double check that because we have a lot of information here. And you can see here RB and B are 3.5. And so that brings up a good point in that these aren't going to always be exactly accurate. And it's complicated, but there's crossing over and different ways that these things happen. It doesn't always turn out exactly perfect. So I'm going to go ahead and make a notation that RB, since it was 16 away from VG and 3.5 here, and that one's probably the most accurate, so I'm going to make a note that that's 3.5. So I think the only one I got to figure out now is CN, and so CN is a distance of 9 from VG. So let's see here. So that would mean probably about right here. So CN is a distance of 9, which means this is a distance of 10 from CN to B. Let's check that. CN to B is a distance of 9, so they're saying it's 9. So what's the distance between CN and RB? CN and RB, oh look at that, 6.5. Alright, 6.5 and 3.5 make 10. So you can see those smaller numbers tend to be accurate. The bigger numbers maybe not as accurate. So they're not going to always mathematically work out, but I'm pretty confident that I got this one straight. So the order of the genes, depending on how they would list it, would be VG, CN, RB, and B. So that's really it. You just got to put the pencil to the paper and play around with the numbers a little bit. Remember to start with the biggest number and then kind of work out the math. It's not too difficult. I think you can figure it out. Good luck. I hope this was helpful.